Well, Richard, thanks so much for taking some time this afternoon to chat. You were a professor at Stanford from 1982 to 2001 before joining Genentech, leading perhaps one of the most important research groups in both industry and academia, developing medicines that literally have touched millions of lives. And so I want to start by asking you, what was that transition like from a distinguished academic career into industry?
Well, the transition was scary, frankly. And, when you're a tenured professor at Stanford, you have a reasonable life. You really can't be fired. I had enough money to do the work I wanted to do, but our research had reached a stage where we had understood some things, and I had to sort of take a step back and think, what do I want to do with the next 10 or 20 years or more? And I was fortunate enough to be contacted by Art Levinson, who asked me to come and talk to him about whether I might want to lead a group at Genentech, which I did. And I thought, wow, what a terrific thing to be able to try and use the instinct that I developed around life science to make medicines to try and help people. But boy, walking in the first day was pretty interesting. I went from a lab of 20 people to eventually a group of 2,000 people, a budget of maybe $1 million to eventually a budget of about $1.6 billion. And I think I owe my success to mentorship. And, academics often move to industry without appropriate mentors. And I was very fortunate to report to Art Levinson, who is a basic scientist himself, and to have not only his mentorship, but the mentorship of Sue Hellman. But mentorship was extremely important to me in making that transition. And I think it's something that people often lack when they move from academia to industry, and that can result in, and has resulted, and we won't go through the details, in what I consider to be some catastrophic failures.
So Richard, it was certainly my experience when we overlapped at Genentech, and it's also the reputation of the research group at Genentech as having an extraordinary culture in terms of how they approach science, particularly in industry. And many companies try and emulate that. I wish it'd be great if you could talk a little bit about how you sort of built that research culture at Genentech to allow people to have goals like we do in industry, but also to do things that people didn't think were possible. Because I think that's so relevant for young companies starting now.
Well, Genentech was fortunate enough to have been successful by the time I arrived. So we had certain freedoms that I don't think a startup company necessarily has. But it was extremely important in the culture at Genentech, such as a postdoc program. We had over 100 postdocs, and their job was to do basic science and to write papers, and then to go off and get jobs the same as a postdoc would do in a university. But these postdocs were positioned next to technicians or Genentech scientists that were doing drug discovery. So it was really the mix of basic science done by the postdocs and more directed drug discovery science done by the Genentech full-time employees that I think gave rise to an atmosphere of very, very rigorous science. And I know people talk about that a lot, rigorous science, and they say how important that is. We do rigorous science. But I think one really has to ask oneself today, how rigorous is life science really? I mean, we found, and Amgen has published papers on this, that approximately half of the papers or half of the conclusions in papers, half of the data in publications is not reproducible. And this gives rise to really difficult issues in industry and startups. If you're going to spend, how much? Tens of millions, sometimes hundreds of millions on a drug discovery program. Holy cow, you better make sure that the data that the program is based on is correct. And much of the time, we found it impossible to reproduce the published data. And I think that's a very serious issue as we go forward. There's a lot of money in science, in particular in life science now. And there's a huge amount of pressure to publish. And I think there's a lower than ever concern about rigor in publishing. So there are real issues with rigor and real issues with maintaining a high level of science in industry that is frankly lacking a bit in academia, because there's really no repercussions for publishing something that's wrong. Often you publish another paper correcting it, so you get two for the price of one. So no, and it's a very serious issue that I know people in academia also recognize and are attempting to grapple with.
One of the things we hear all the time, how much truth there is hard to know, that in some big companies, scientists sometimes want to avoid doing what could be the killer experiment because they want to keep their project going, because they don't want to lose their job or lose their funding for their group. So how do you create a scientific culture where people are incentivized to do the killer experiments early and reward people for getting an answer, not necessarily an answer that keeps the project going?
Well, there's two ways, really. I mean, number one, it always catches up with you. So, fine, you can maybe scrape out an extra year or two of some kind of funding or glory or what have you, but it's going to catch up with you in the long run. But we also had, and Genentech still has, very, very active review of the programs by the most senior scientists in the company. So, high level review by the most rigorous people in the company that are not as emotionally attached to the programs, we think, resulted in better overall science. And, to be fair, I suppose it was scary on the one hand. On the other hand, we provided, written minutes and action items after each presentation, but we fully acknowledge that the scientists presenting the data better understand it more than we do. And if the scientist disagreed, it was perfectly legitimate for the scientists to come back and correct the mistakes.
Yeah, so as you know, both of us are now spending a lot of time trying to help young companies get started, working with founders and their first few employees. What kind of advice do you have for them as they're building that first 20 employee base, which are likely going to all be scientists?
Well, I have two things, that one of which I already mentioned, mentorship. And it can really be incredibly valuable. We're graybeards, which is a polite way of saying something. And, we've made a lot of mistakes in our careers. And, we've also done a few things that weren't mistakes. But, boy, to be able to help a young CEO or a young chief scientific officer by saying, boy, back, 15 years ago, I faced a similar situation, and maybe you could consider, this or that, can really, really be extremely helpful. So I really suggest that people look for mentors and people be open to mentorship. number one. And then number two, the other thing that I look for is that it's great to be a visionary. And, we on the one hand, we're looking for visionaries. On the other hand, you have to have a sense of practicality to go along with the vision if you want to be successful. So, I like a down-to-earth visionary, if there is such a thing, because you can get too caught up in a vision, and before you know it, you've spent a lot of money and your company's pretty far along, and maybe not a Google, but somebody will be asking you, where's the product? where's the beef here? And, if you don't have it, you're going to be in trouble.
So let's shift gears. So you left Genentech, you became the chief scientific officer of 23andMe, and you helped establish their drug development program, taking its genetic database and trying to turn that into a resource for new therapeutic targets. And we all know that genetics are powerful predictors and starting points in drug discovery in helping to select the right patients that may benefit from a therapy. And so you walked into 23andMe, which was a commercial company offering testing for millions and millions of people around the world, never had done therapeutics. How did you approach it? And how did you think about the potential impact it could have on the way we think about drug discovery today?
We started out not really even speaking the same language as the geneticists. That was funny. Someone I remember came to me and said, oh, we have a really interesting genetic signal in, I forget what disease it was. And, it looks like a really strong signal. And I said, oh, well, what gene is it in? What protein is it in? And they kind of said to me, well, is that important? And yeah, that's kind of important for where I want to go. So, we had to develop a common language between the statisticians and the statistical geneticists in order to, really begin to talk seriously about drug development. I will say that the database was it still is, absolutely fascinating. It's not 12 million people. I don't know if it's 3 or 4 billion phenotypic data points. One of the things that we ask is whether you're a morning person or an evening person. And that question leads you to the circadian rhythm genes, which is, we don't even define what a morning person is. And you go right to the period gene. You go right to the clock genes that determine our circadian rhythm. So, it's absolutely fascinating. And I do believe is a terrific way to guide drug development. I would say, though, that it's very different than oncology. And there, one needs to be careful. When you have a somatic mutation in a gene that drives the tumor, we've seen, and you know more about this than I do, that, drugs against those drivers can be extremely effective and have a very high probability of technical success if you can make the right therapeutic agent. The biology is pretty clear. For complex disorders that are polygenic, a genetic hit really only gives you a clue about something that may be important as a drug target or may not. You really have to understand much more about the biology. So the connection to the disease is, I guess it might be fair to say, somewhat less direct than an oncogenic driver. So there's a lot more biology that needs to come into play, a lot more thought, perhaps a lot more preliminary experimentation around a therapeutic hypothesis that comes from the polygenic disorders versus oncology. So we're all, for the experts, we're all looking for the next PCSK9, and I have news for you, there aren't very many. So, but having said that, I do believe that, we have, and many people have, by following a human genetic approach, uncovered a number of targets with great promise. The other thing that's fascinating nowadays is to not only target particular mutations with clinical trials, but to, in general, imagine if you could develop a polygenic risk score in a particular disease such that it was extremely likely that the people would show symptoms of the disease, years faster than someone in the general population. So if one could then enroll these people in your clinical trials, you would be able to see a signal much faster with a much lower number of folks in the trial and do drug discovery much more efficiently and much faster. So there are many uses of the human genetics that I think we're just beginning to experience.
Certainly in oncology, we've developed, starting with Genentech's development of Herceptin, precision medicines where we only enroll patients where we know the target, the drug only hits that target in that subset of cancer patients, and we see the effect. And we're all just waiting to see how this will play out in other therapeutic areas, particularly your area of basic research in neuroscience, because right now we know that we're treating everybody as one disease, but we know that's not likely biologically what's going on. And so
yeah,
we
and
get there.
I'm sorry to say, I'm seriously sorry to say, it's not going to be as straightforward as oncology for the reasons that we just discussed. I think it's much less the case that there will be equivalence of oncogenic drivers for those complex diseases.
Yeah.
Now, which doesn't mean that it isn't exciting and doesn't mean that we won't make progress, but it does mean that it is going to be harder, slower, and more expensive.
So that's a good segue to a discussion around how do you think drug discovery within companies, large or small, is going to or needs to evolve? Do we just keep going the same way we've been going? Productivity rate has not been amazing. Or do we need some sort of shift? A lot of people are talking about how machine learning is going to do that, or genetics. But where do you think we are in that?
Well, I think the shift is taking place naturally as the science matures. If you look back, and these statements will be generalizations, which someone can always find exceptions to, but let's look back 30 years or even more. What a number of us were interested in then is defining the genome. What is the human genome? How big is it? What's the sequence basically? How many genes are there? What are the genes? And we've largely accomplished that. We know the human genome, we know approximately how many genes there are, etc. Then we move to the stage of saying, okay, now that we know the genome, what does the genome do? What does gene X do? How does it function? What role does it play in cell biology? And what roles might the gene play in various diseases? And, over the last, I'd say, then 20 years, we've made terrific progress on that. There's still a lot to be done there. But now we're also moving to the era of, once we know what a gene does, in quotes, in the broadest sense, and that it might play a role in a particular disease, we're asking the question, how does it do that? And that's a chemistry, biochemistry, or even taken to its fullest extent, a physics problem in a way. So, what the life scientists are trying to do is to take biology and make it a chemistry problem. And, we've made pretty damn impressive progress on that over the last decades. So I think that puts a drug company in a terrific position, because that's what you don't want to have to figure out what a gene is, what a gene does, how it works. You have all that now as your substrate to work with. And that is really, really exciting. Certainly I am a little worried that there's almost too much money going into startups nowadays, and that we're potentially a little overheated. I was in a discussion with people that I really respect earlier this week, who have said that there's something like $1.5 to $2 trillion sitting on the sidelines that needs to be invested in some way other than government bonds, which gives you no interest. And certainly some amount, a significant amount of that money will likely go into biotech. And I think the fact that that money's there has resulted in the recent large number of ATMs, a large number of SPACs. I mean, it comes back to the issue that I discussed a little bit before of what is the real solid science? What's the science that makes sense? And, can we really figure out where to invest in a thoughtful way versus scattering money broadly, some of which will be, perhaps too much of which will be wasted versus having thought about the problem more deeply.
I'm curious, what particular areas of science or medicine are you most excited by right now?
I remain very excited about neuroscience. However, I do think it is not going to be filled with short-term returns, and that it's going to be a long, hard slog. It's just such an intellectually fascinating field of trying to understand the basics of how we think, what it is to be conscious, and then what happens in, for example, what will certainly be the next pandemic of our times, dementia, and how that feeling, how that recognition of self, how that ability to think, declines as folks lose cognition. And what we can do to slow down that decline is, I think, intellectually, probably the most interesting challenge in the life sciences. But, I caution, an extremely, extremely difficult one.
We really appreciate you taking the time to talk to us today.
Pleasure.
Thank you, Richard.